iodine test is a simple yet powerful biochemical assay. It's used to detect starch and related polysaccharides. To perform the iodine test on bacteria, begin by streaking strains onto a starch agar plate. Incubate the plate at 37 degrees Celsius for 24 to 48 hours. This allows bacterial growth and possible starch hydrolysis. After incubation, flood the surface of the agar plate with Lugol's iodine, which contains 5% iodine and 10% potassium iodide. Make sure the coverage is even and let the iodine react for 30 to 60 seconds. Now examine the plate for color changes. A deep blue to blue-black coloration indicates that the starch is intact, meaning the bacterium does not produce amylase. A clear or light yellow zone around the colony indicates starch hydrolysis, confirming amylase production. The iodine test can also be performed directly in solution. This method is especially useful for food samples, biochemical preparations, or plant extracts. Place 1 milliliter of the sample in a clean, dry test tube and prepare a control tube with 1 milliliter of distilled water. Add 2 to 3 drops of Lugol's iodine to both tubes and mix gently, then observe the immediate color change. Whether applied on agar or in solution, the iodine test provides valuable insights into starch metabolism and polysaccharide composition. The iodine test works because polyiodide insert themselves into the helical structure of certain polysaccharides, stabilizing the helix and producing characteristic colors depending on the polymer. Amylose, the linear form of starch, produces a deep blue. Amylopectin, which is branched, produces a reddish-brown to orange-brown color. Glycogen, which is highly branched, gives a weaker reddish-brown reaction. Simple sugars do not react, so the solution remains the natural yellow-brown color of iodine. To distinguish weak reactions from the natural color of iodine, remember that natural iodine is yellow-brown and transparent. Weak positives, such as glycogen or amylopectin, appear reddish-brown, darker, and more opaque. Always compare your sample with a negative control to ensure accurate interpretation. As with many microbiology assays, the iodine test has limitations. Misinterpretation is possible if certain errors occur. Uneven application of iodine can lead to inconsistent color development. This test cannot be performed under acidic conditions, as starch will hydrolyze under such circumstances. Additionally, the iodine test is a qualitative assay. It indicates the presence or absence of starch, but does not provide information about starch concentration.